number of institutes that are participating in the Blueprint Neurotherapeutics Network, and they are listed at the bottom of the slide. And my email address is here. It'll show up again at the end of the talk. So fear not, if you don't get a chance to write it down here, you will get it. And everybody here will get a copy of the slides via PDF of the slides when, after the talk. Rakonda will send those out if you're participating today. All right. So the Division of Translational Research at the National Institute of Neurological Disorders and Stroke, NINDS, has a goal to accelerate basic research findings towards patient use for neurological disorders and stroke. And we have an array of funding opportunities to make that happen. And some of those are trans-NIH, like this Blueprint Neurotherapeutics Network, and others are focused on NINDS mission grant funding only. But we also cover, the whole spectrum of in, indication areas in terms of NINDS, as well as therapeutic modalities, um, biologics, small molecules, and devices. I will focus on small molecules today, but know there are other funding mechanisms out there for these other modalities. So let me introduce you to the Blueprint Neurotherapeutics Network. It is what's known as a cooperative agreement. And as a cooperative agreement, that means that there is substantial NIH interaction on your program after it gets funded. And we'll talk a bit today about what that looks like. And know that there are two funding opportunities. One is a SBIR version, and the other is a non-SBIR version. So if you're an SBIR eligible organization, you can consider the U44 version of the program, but know that any small business, SBIR or otherwise, is also eligible to apply to the UG3, UH3. And sometimes there's reasons why SBIR eligible groups do go to the non-SBIR, and we can talk about that a bit later as well. As I mentioned, the program does have a biologics co-part to it. I'm not going to talk about those today, but I will give you the funding opportunities later. The next application due date is July 15th. We have these twice a year, typically in July and January now. So what is our vision? Combine the strengths of NIH with industry expertise in small molecule neuroscience drug discovery and development. We rely on you to bring your ideas to us with novel drug targets and strong disease assay models. And we can provide all the disease agnostic expertise required. So we have advisors with extensive pharma experience and industry standard contract services, and I'll go into more of that in a moment. It was part of a grand challenge of NIH Blueprint for Neuroscience Research, and as I said, the goal is to overall de-risk the program that you bring in to a point where industry will invest in it and take it farther and down the development pipeline. We really want to see the programs progress towards registration, helping patients. And so we are looking for that handoff or getting it ready for that next investment stage. The idea is to identify the best ideas for translation that we can fund and provide non-dilutive grant funding and resources to move the project forward. One of the hallmarks of this program is we're trying to preserve your institution's intellectual property to facilitate this licensing. I might show this again, but the entry criteria is a minimum. You have a small molecule to optimize, and you have the SAR driving assays available, at least the in vitro SAR driving available at year one, and animal models known and can be incorporated in your lab or through a collaborator. You can enter anywhere along the pipeline, and that's why it could matter if you're coming in at a later stage. You might need to have those animal models in place, ready to go on day one, and in other cases, it may not be required till later in the program. Um, we can take you all the way through, no matter what. The goal is to go all the way to phase 1 clinical testing. So the grants are always written in terms of going all the way to the IND filing. And typically grants are that we have in ours are going into clinical trials through our contract side. But it is possible to do clinical trial uh, on as part of the grant. It just requires some additional hurdles. External funding and partnership, as I said, is the key goal for getting the long-term funding for phase 2 and 3. But sometimes that's not possible in the end, and the exit is actually to other grants. And obviously, it's a milestone-driven program. Attrition can happen for any number of scientific reasons along the way. Um, so here's an overview, a sort of a snapshot of what the program looks like in more detail. We have, as I mentioned, substantial NIH input. Our interaction, we work with you, meet every 2 weeks in what we call the lead development team. And the key contributors to those meetings are the principal investigator and their team, the industry consultants that we assign to the project, in the various disciplines, depending on the stage of the program. You will eventually go through all of these disciplines, but you may start with just medicinal chemistry, for instance, or in PK, or maybe some assay development in medicinal chemistry. It depends on where you're coming in. As I said, you have to have at a minimum, the bioactivity assays for the disease biology, and that's because we have hundreds of diseases, and we just can't provide you a contract that can do all of the disease biology. So that needs to be part of your grant budget. Um, we will custom tailor the contracts to meet your needs. So if you don't need everything, um, we can do parts of this and not everything. Um, these are the contractors that are presently in our pool for doing different things. Medicinal chemistry is Curia. Drug manufacturing and formulation is Curia and MRI Global. PK and Tox, we have 4 different vendors that do work in that area. Clinical trials, we have PPD and Duke. And if you're doing optimization work, we have a relational database system, CDD, to help you. And what you really have to ask yourself is, do you have the expertise in-house and readily available to do these activities? And one of the key things I always run into is, we'd like to do, for instance, medicinal chemistry in-house. And so the question I'm going to ask when that happens is, do you have the chemist ready on day 1? Because the milestone starts on day 1 um, of, of the grant. And are you ready, if you miss your milestone, do you have an easy way to shift them to somewhere else? Or are you going to be forced to fire those people if you were to miss a milestone in the project started? That's why many people choose to use our contractors rather than do this. That doesn't mean you can't have support from medicinal chemistry, your historical um, collaborators work with you on this. You just have to be clear to um, in the grant who's doing what, and it calls for tables in the grant to actually delineate what you plan to do yourself and what you're asking us to do. I highly recommend, and I'll say this multiple times a day, if you have questions about this, please, please, please contact us before you file your application, to make sure we understand what you're looking for and that we can give you good advice on what's going on. Just keep in mind, this section is NIH contracts. They are not in your grant budget. And so none of this is budgeted in your grant, and none of it goes towards the limits that are associated with the grant funding. Here are the contacts for all the different ICs that are participating presently, institutes and centers, and they're listed in the end of the funding opportunities as well, along with their phone numbers. And so you don't have to write this down. I just put it here to show you who, in fact, is presently part of our program and is supporting new projects within it. As I mentioned, it's a milestone-driven program, but it's also a phase mechanism. It's a UG3, UH3, or a fast track U44, where there's a decision point, typically after 1 year, for always in that case in UG3. Um, Sometimes there's a reason that you can go 2 years for phase one because the rules in SBIR um, only allow a 3-year phase two. And so some people spread the phase one over 2 years to have a 5-year total. The UG3 UH3 mechanism is often 5 years if you're starting exploratory. Again, it could be less if you're coming in later on the discovery pathway. Um, Again, if you're coming in, we'll want to talk to us about what would go into that preparatory phase, and make sure you have clear idea of how to properly spread the work, and understand the speed at which we do stuff and the parallel versus linear nature um, that you will face in some aspects of the program. Um, so that being said, we will work with y'all on a set of milestones. Our milestones are in our funding opportunity announcements out there. Um, uh, Both of them have a link to the BPN milestone orientation in there. It's also on our website in frequently asked questions. And you can see what exactly we have for milestones based on stage. For most projects, there may be additional ones specific to your project, but the overall disease agnostic type of drug discovery milestones will be there. And you can see how the progression, what resources are available at each stage from the contract side. Each project can only receive one UG3 award. I get this question as well. Fairly often is, can I have, another UG3? And the answer is no. We do a preparatory phase, we make a decision on the milestone, and there are milestones every year of the program. So we'll either progress you to UH3 or we'll stop at the end of UH3 or UG3 and not progress. And that's true even as we go in farther years down the road. We'll look at the grants and make a determination whether we're going to go forward. The execution period, the UH3 or the U44 phase two, typically supports MedChem in total up to 3 years. So it says 2 years here, 1 year here. Um, so it depends again on your entry point. Be careful, talk to us, and we'll make sure we understand what you're trying to do. But make sure you go all the way to the end. I do want to also point out that not all of the ICs that are participating accept development projects. So coming in and say, I don't want to do any more optimization. Here's the compound I have. I need to finish all the data set for that compound, and then I want to move into development on it. If you're at that stage for NIDA projects, drug abuse, or mental health projects, you really need to talk to them. They have other opportunities they would prefer you use for this. I have budget information down here about it. And for the UG3, UH3, these are guidelines that give you an idea of what we typically see. But your particular grant should reflect the actual needs that you have. And it should not exceed $499,999 in any year of the grant, unless you have gotten prior permission to come in with that. And that takes 6 to 8 weeks prior to receipt date. So if you're planning on coming in the July date and you plan to exceed direct costs of $499,999, then you need to send me very quickly a set of specific aims, a very detailed budget, and we need to have a discussion about it. And we'll talk more. The U44 stage programs don't have that limitation. They have different set of rules that are based on the IC. I have NINDS's rules here, which has up to 500K in phase one, 700K across if it was 2 years in total cost, not direct costs. So your cost of your indirect plus your direct plus your fee, your total cost, goes into that number. And in phase two, up to $1.5 million per year, but total of 3 million across the maximum of 3 years. So that limits you in that regard. So you just have to be aware of that in time. You may have less time in this scenario, but you have an easier access for coming in with higher budgets in the later stages, and you can charge a fee. So those are the advantages of it. The disadvantages can be related to that 2-year time frame um, in phase one where you have less money, and it can be also a consideration for how good is your plan for everything in there. You'll be restricted for if you need a foreign CRO, for instance, to do the work. You can't do that at SBIR. If you have a collaborator there, well, you can do that here. So some small businesses, due to their actual design of their program, end up being in the UG3, UH3 because it's preferential for the overall program for them. I highly recommend you talk to us again if you have questions about this in your particular project. Um, Applications from foreign institutions, just keep in mind, if you're coming in as a foreign institution, there's 2 levels of review. There's the first level, which is just the standard review, and they will comment on the foreign um, applications, but they also have to go to council review, and they're looking at does this grant offer unusual talents and resources populations or environmental conditions that simply don't exist in the U.S. readily or augment existing U.S. resources substantially. So you'll have to make that case as well in your application. We talk about entry points, whether it's discovery or development, as I said. Medicinal chemistry, if you're still doing optimization work, you're in the discovery phase. But I will say almost nobody comes in truly in the development phase um, for if you look at our milestones. Most people are coming in at if they're doing optimization, they're clearly discovery. If they're coming in development, they're most typically at the end of lead optimization, where they're not asking for any more um, optimization work, but they are asking for things like dose range finding tox or um, additional PK work in large animals or any number of things that we would normally have by the time we say it's a development candidate, as well as a whole profiling of the in vitro admin, things that many people just don't have a full package. So if you're coming at this stage and you don't have the full package, what you're really saying is I'm coming at the very end of discovery and my preparatory phase is going to be gathering all that remaining data and then making a decision, is this truly a development candidate at the end of the preparatory phase? So think of it in those terms. If you have a fantastic set of data, it's fully fleshed out, then you can potentially come as and declare yourself as a true development candidate. But keep that in mind. This is just sort of an overview of the milestones I've been talking about. In the early exploratory phase, it's about does the assay work? Do we have a screening funnel and target product profile in place? And does the medicinal chemistry make sense? So if you say I have some hits, we may resynthesize those hits. We may do some limited SAR. We will do things like early in vitro ADME profiling. We may do in vivo PK on the best molecule just to figure out what the liabilities are, and do we have a data-driven plan to move into optimization full force and hit to lead and lead optimization. As you go through hit to lead, things progress from making multiple series potentially down to one series, picking your candidate, as I said, getting the PK/PD and rodent DRFs, and then eventually moving into large animal dose range finding, CMC starting work, and make sure you have clinical specialists. If you can complete all that, then you are ready for the IND enabling studies in the downstream um, clinical trials to come if everything works out. The grant duration is a maximum of 5 years for the combination of preparatory and execution phases, no matter what we do. Intellectual property expectations, I mentioned it at the beginning. I'm going to reiterate it here. Inventorship is always determined by U.S. patent law. There's nothing we can do about that. So your collaboration with our consultants and our contractors means that you will have inventors that are not part of the institution. But Blueprint is not seeking any stake from the NIH side in what you're doing, and we set up our contracts that way so that prior to the grant, we must have IP agreements in place for those internal collaborators you have, that you have worked out your expectations, who's the lead for intellectual property, which institution. And likewise, we will set up agreements with our consultants that deal with assigning the IP to your institution, and any issue of, what the if there's any kind of royalties or not, you put that in with your, and it's typically in your only internal folks are doing that. IP agreements should aim for unencumbered IP consistent with the program's goals to create a licensable project at the end. I mentioned the website earlier. This is the link for the website. And you can see, if you were to click up here, you could change from small molecules to the devices, to the biologics are all listed here. And you can switch between the websites. There's funding opportunities listed here. There's a wealth of information, what our current projects are, how the model works, who was on our external oversight committee, what resources, those are talking about the contractors and other things that are available through us with more detail, who all our consultants are all listed there. The frequently asked questions, there's a lot of them, and one of which is the how do you build your milestone plan. I really highly recommend you look through the site if you're going to apply to the program. There's a lot of information on it. The program itself, just, it's been around for a little over 10 years. I think we finished 12 at this point, and has had good success. The programs have put a number of things into the clinic, and the ones we have put in the clinic are progressing. So there are 4 projects now that have progressed from the early stage of the programs into phase 3, and some newer entries into phase 3 now from the middle side of the program. And then, we hope that your new projects will be the future um, graphics on this as we move things forward. 10 of our projects have already announced additional funding, and that funding, I will say, is potentially in excess of $3 billion. So it's not insubstantial in terms of investment they have. You have projects that have been licensed to major pharma, projects that have been part of major venture programs, and you've had things that have moved with, large pharma in Japan, as well as other venture capital. So there's a lot of different kinds of investments in the programs. So let's just say you don't have everything you need to move forward yet. What do you do? Um, there are programs that we have at NINDS, for instance, program called Ignite, uh, is designed to get you additional data. Those data could potentially include things like assay development and running a screen. It could talk about developing an animal model. It could talk about, I have that, that quote development compound or my major tool compound, and I really need more robust um, efficacy studies to really build up what I, need for preliminary data to move into another program like BPN. These are stage programs. They're maximum 750,000 direct cost. There's no SBIR version of it, to my knowledge, but they're extremely clear, quantitative, and definitive milestones are an essential part of it. It has one go, no go decision point that takes you from R61 to R33. But these are our programs. So you set up the milestones beginning, it's agreed to, and you're not meeting with the NIH all the time like you are with BPN. There's no additional NIH resources. This is you and your group moving things forward. There's also a regular training program we have every year, run presently through a grant to UC Davis for drug development course in drug discovery and development for academic science. I point this out is there are lectures in all the different disciplines like medchem, like PK, like tox. So, if you need a primer on sort of what goes into tox and what considerations you need to help make it potentially a better application, there's course material on here for all of those, a quick hour of your time to get a primer on what you should be thinking about for tox or PK or anything in the drug discovery realm is in these lectures. And this lecture series covers not only small molecules but also biologics now. They'll be accepting applications for next year's class in 2025. We'll probably towards the end of the year. You can scan this code to get more information or contact Teresa. So I always like to end the webinar with some things to do and some things not to do. So some things to do. Please read the funding opportunity very carefully. Do discuss your proposal with NIH. You know, I'm here available to discuss it. And if it's not going to be an NINDS application, those contacts I gave you, or within NIS missions, those contacts I gave you and the other ICs will be important to loop in as well. So you can start with me, and if we can discuss if we need additional IC input, we can either have them on the call the first time, or I can give you the follow up after a discussion where we have how we do it. If you are thinking about doing indication areas that cross institutes, then you really need to think about which one you have the best data for, because it's difficult to get funding from two institutes simultaneously. If you're doing SBIR work, and you have SBIR questions, you need to contact the SBIR program director in the IC that you are thinking about doing, because they have different priorities, they have different funding limits, um, and you need to really know what they are for that IC. Stick to the page limit. If you don't, they will withdraw your application. It'll never get reviewed. Make sure you have solid preliminary data that supports your proposal. Address the rigor of that data in your research strategy section. I can't emphasize this enough. In the research strategy section, not the vertebrate animal section, the research strategy section. That's where the reviewers are going to be looking for you to address rigor. If you do not put it in this section, they will not give you credit for it being somewhere else, and you will get dinged. Address the obvious criticisms of the chemistry and biology. If you don't, the reviewers will assume you just don't recognize that as an issue. So if your compound has a structure alert and you think it's obvious, we're going to get rid of this. I put it in my plans. I would call out specifically and say, I recognize there's a structural alert, and that's what I'm trying to get rid of in my optimization strategy. And I believe I can because of this, this, and this. Clearly indicate what's going to be done as part of the grant and what you're expecting for BPN contracts. It actually calls for this in the funding opportunity announcement. Put a table in that says it. Please do that. This one I get at least one or two applications every round. And unfortunately, some people submit theirs on the very last minute and I can't get them to correct it. I have to have grant years for every year of the grant, from where you're entering all the way to the IND filing at a minimum. That needs to be in there. So please make sure you do that. Make sure you explicitly discuss the data quality, as I mentioned, and the rigor and the data management plan as required in the funding opportunities. Look carefully at the funding opportunities. The languages change fairly regularly as NIH updates different aspects of it. So you just make sure you're looking at the latest version of the funding opportunity and that you're following all the rules associated with that. Complete your registrations at least 6 to 8 weeks in advance. Again, I get this fairly regularly at the last second. You know, I can't get it registered. Sam hasn't responded to me. And sometimes it really does take 6 to 8 weeks. Some people are lucky and it goes a little faster, but you should give yourself at least the 6 to 8 weeks, which means if you're planning on doing the July date and your institution or you yourself as a PI don't have your registrations done, start today and start getting that done. Consider putting your application in early and letting me know it's in when you've done it, because that gives me opportunities to at least look at it and say, do I see any obvious things missing? I can't rewrite your application for you, but I can let you know that, hey, this is missing, like the budget sheets, or like I see an issue that, you know, there's some issue somewhere in the grant that I think you may have to address. Please address it. And you can have an opportunity to withdraw the application, put it back in with the corrected version. Um, if you're applying to the UG3 UH3 program and your budget exceeds, the 499.99, not 500 as it says here, but 499.99, then you need to ask permission to submit, and request must be made 6 to 8 weeks in advance. So again, soon. Um, some institutions can move it a little faster, others cannot. So please don't wait. Get it done early. I would highly recommend you have a conversation with your tech transfer folks or your business development person about how you're going to prosecute patents and licensing activities, because we're going to be asking about it, and it's going to be important to make it clear to the reviewers that you have a plan. It calls for intellectual property plan in the UG3, UH3, and it calls for commercialization plan in U44. Things not to do. Do not plan on this being your sole funding for your lab, because it is milestone driven and can end abruptly. We don't ever want to see you have to, get rid of people because of that. So don't plan on this being your sole funding for your lab. Please do not under-resource your budget to avoid limits, because coming back later and saying, well, I need more money, may or may not be possible then. I would rather get the ARA done if in fact you know you need more money, and you can justify it up front, than to you try to go under it. It's not, that doesn't mean, you can't have alternate funding that you're augmenting this funding with. And sometimes that is just fine, and people do that. They have more than one stream of money, and that allows some stuff to actually happen in parallel when you have a second stream of money helping on this. Do not plan for BPN contracts, as I mentioned, for disease biology, or for that matter, for one-off in vitro admin experiments. If you just need to run SIP phenotyping, and that's all you need, then put that as part of your budget. Because for me to put a large contract thing to do one experiment is a lot, it ends up costing a lot more than if you just put it in yours in the first place. And it'll be faster if you do it that way. If you need a whole in vitro ADME support for the program, that should come to our contractors in most cases. Do not code your application as a clinical trial if you plan is to use BPN contractors at the end, because the contract is a clinical trial contract, but the grant is not, right? Because there's no funding for going to the clinical work in the grant itself. So that's the demarcation there. If you plan to run a clinical trial, it most likely will be a delayed onset because you don't have all the information yet to do it. And so again, you will have to put information in, but it's most likely will be a delayed onset. I promised I would give you the biologics funding opportunity announcements. Here they are, and the contact is Chris Boshoff. Here they have some slight differences in the program. Their contracts, they use some of our contracts, but they have their own suite of contracts because they have multiple modalities. Those contracts are set up a little bit differently, but the goal is still protecting intellectual property. They have potentially a 2-year UG3 in these, because some of the biologics require a longer period to get things done in the earlier stages. So that's very similar, same rules. Not everybody accepts development projects. We also have a companion program that does both small molecules and biologics. It was based off of the Blueprint Neurotherapeutics Network that I launched and is now run by Marianne Pelamounter, that is focused on pain therapeutics development. So the HEAL, Helping to End Addiction Long-Term Therapeutics Development Program. So if you're planning on doing pain indication, you really should consider this program as your first choice. It's an RFA. It has a set aside separate pool of money associated with doing pain indications. The program is set up very similarly. The major difference is it does take biologics and small molecules under one umbrella. It also is looking for you to do some pharmacodynamic or target engagement type biomarker work as part of this program as well. It's eligible for small businesses and academics. There is no U44 version, no small business version of this, Marianne Pellemounter is the contact again. So at this point, I'm going to transition to questions, and those questions can be related to the application process, to the review process, to proposals and your budgets, contracts, intellectual property, any of the above. And as promised, my website and my email address is here, available for you in the future. So at this point, I'll move to questions.
Thanks, Chuck. We have just a couple, and for those that joined late, you can submit your questions in the Q&A chat section below. First question, hello, can the small molecules be the FDA approved compounds or must be brand new ones?
I will say repurposing of FDA approved compounds is a very challenging thing from a number of different areas. Um, it will depend on the discussion, but commercialization plans, intellectual property plans related to someone else's compound are always very challenging, as is investment in those down the road to get them beyond phase one, into the clinic. There's also a question is, will this be, there's a caveat to the program, anything directed beyond phase one is out of scope. So if you're really doing work to enable a phase two clinical trial, then you're probably out of scope. But as they said, the devil's always in the details. Happy to talk to you more about it.
Okay, the next question, at what stage are your BPM projects licensed to major pharma?
Um, at all different stages, I'll be honest. We have projects that were licensed to pharma after they left the program. We've had projects licensed to pharma as early as hit to lead. The most typical inflection point is they will make a licensing agreement and then take it over after our single ascending dose first in human clinical trial. So sometimes they come in and work, just join the LDT. They may have additional things they're doing in parallel at the licensor, um, licensee, sorry, site to help them or to do additional work that they would want that's beyond what we would do. Um, and so it's really a collaborative thing. But sometimes they just license it and take the whole program. And I've had that too. Say, okay, we're happy. Like I said, I've had people license as early as hit to lead. I've had people license at programs out at essentially before after GLP talks, before scale up for GMP. I've had people license it after the clinical stuff is over.
All right, next question. What if a compound has already been patent and in vivo work performed? Can we still apply for this mechanism?
One more time, I didn't quite catch that.
What if a compound has already been
Patented.
patented and in vivo work performed? Can we still apply for this mechanism?
If you're looking to do the IND enabling work, then yes. If you're just looking to do a clinical trial, then no. Because there are other funding opportunities for just doing a clinical trial.
Okay, will you fund a MAD study? If so, would you do a 28 study?
A 28-day study. We
Yes.
typically do 28-day GLP tox as a routine, with a typical reversal of 28 days. That's our pretty standard GLP tox repeat dosing studies. We don't typically fund the MAD study, I'll be honest, in most of the programs. The HEAL program for pain, I think, because that's a separate stream of money, will do that more. It just comes down to the cost of those studies, and the inflection point for most investment is after the human PK is really where they're looking to make the investment.
Okay, next question. Would RNA drugs such as microRNA be a fit for this program?
We need more details about what you're doing. It would be a fit for BPN. It might be a better fit for the BPN biologics than the small molecules, depending on how you're structuring what work you need to get done. Um, Small molecule RNA modulation is certainly within the scope, but if it's sort of larger RNA itself pieces, then from a contractual point of view, the contracts and the biologics may be better suited to be able to go out and get sort of experts in that area for work.
Okay, no more other questions at this time. Someone
Yeah.
asked for the slides. Yes, the slides will be sent out in a couple of days.
All right. Well, thank you all for attending. I really appreciate your time. Um, and for those maybe listening on this in the future on YouTube, again, feel free to reach out to me via email. And Rakonda manages my schedule in the world, so she's the one that always figures out how to get you on my calendar as quickly as possible. So thank you, and really appreciate your time. Have a great day.
Thanks, everyone. Have a good day.